Let's take a look at how each of the types of organic molecules moves through the digestive system and is broken down into monomers that are absorbed and then can be used by the body. We'll start with proteins, which proteins, because we don't have any enzyme in the oral cavity, remain whole in the oral cavity and in the esophagus. But once we swallow and the protein reaches the stomach has pepsin, which was converted from pepsinogen by hydrochloric acid, and it begins protein digestion in the stomach. Remember also that the hydrochloric acid is denaturing the proteins from our food so that the pepsin has even better access. Okay, so the after the we're finished in the stomach, the pieces of protein that have been digested by the pepsin move into the small intestine. In the small intestine, we have trypsinogen, chymotrypsinogen, and procarboxypeptidase that are coming in from the pancreas. And as soon as they enter the duodenum, the enterokinase, which is secreted by this part of the intestine, activates the trypsinogen to become trypsin, and it activates everything else. So the activated enzymes begin to digest what the pepsin didn't finish. And now we have even smaller oligopeptides. Now that they are small enough, they can interact with the microvilli on the intestinal cells which do what's called contact digestion, because they're contacting the surface of the cells, the membrane, and the enzymes there, which are things like aminopeptidase or dipeptidase, complete the last step of digestion, and now these nutrients can be taken up by apical transporters brought into the cell. Then basolateral transporters move those into the interstitial fluid, which then moves to the capillary, which carries the amino acids to the liver, where they can be sorted, stored, or passed on to cells that need them. Carbohydrates are slightly different because we have salivary amylase in the oral cavity. So carbohydrate digestion begins right away. Not for very long because we're not chewing for that long, but it starts. Now, whatever is broken down by salivary amylase gets swallowed, and as it moves into the stomach, the amylase is deactivated by the stomach acid. So digestion stops until the chyme moves into the small intestine. Here, the breakdown products of the salivary amylase are acted on by pancreatic amylase. And the pancreatic amylase, remember, was inactive in the pancreas because of all that bicarbonate denaturing it. But now that it's in a neutral environment, it refolds and functions. This is actually really important, this idea, because it prevents the enzymes from digesting the pancreas itself. Now, after the breakdown products have gotten small enough, they bump into the enzymes on the surface of the intestinal cells for more contact digestion. And these are enzymes with names like maltase, dextrinase, glucoamylase. They're on the microvilli. They break down the carbohydrates into the individual glucose and fructose monomers, and those are transported across the apical membrane into the cell, across the basolateral membrane into the interstitial fluid, then into the capillary, and then on to the liver where they can get processed as well. Nucleic acids are probably the simplest of all because nothing happens in the mouth or the esophagus or the stomach. All of that basically begins when we enter the small intestine, and the pancreatic deoxyribonuclease and ribonuclease enter the duodenum from the pancreas, and they're also just now becoming activated. And as they break down the polymers into smaller and smaller sections, eventually they're small enough to interact with the surface enzymes for contact digestion, and things like nucleases and phosphatases split the nucleotides into their component sugars and nitrogenous bases and phosphates. And each one of these is transported through its own transporters into the intestinal cells, across the apical membrane, and then across the basolateral membrane to the blood, to the liver. There's a pattern here, which we're about to break, because now we're going to talk about lipids. Lipids, even though the lingual lipase is secreted in the oral cavity, remember it doesn't become active until the stomach, where the pH is acidic. And the lingual lipase begins to break some of the triglycerides into fatty acids and monoglycerides. However, it's problematic because there's not enough surface area. So as we move from the stomach into the small intestine, the bile acids from the liver enter the small intestine and emulsify these giant lipid globs. And that allows the pancreatic lipase to do digestion of the triglycerides even more efficiently. And once these lipids are released, now we have a problem of getting them. One, they're not very happy being in all of that water. They're made a little happier because of the bile acids, but how do we get them into another water environment? So in order to get them into the cells, the lipids first have to kind of be packaged into these micelles, which are a form of the bile acid conglomerate, basically. So these micelles now, which are like little vesicles, can be more easily taken up by the intestinal cells. And they're converted to another version of this vesicle known as a chylomicron. Chylomicrons enable these fats to be transported out of the intestinal cells, but instead of going into the blood, another aqueous environment, they first go to the lymphatic vessel, which is much kinder to lipids, and that lymphatic vessel eventually dumps all of the chylomicrons into the blood, and then the blood takes them to the liver, where they can be processed. So it's the same basic pathway for each of them. The differences are where enzymes are active, and then especially for lipids, how it leaves the intestine.